So starting with a new script. Initializing everything. Okay, now we are going to use fmincon to solve for optimization problems with constraints. It could be equality constraint, and it also could be inequality constraints. To start with this, I recommend you to check the help and type in fmincon, because it helps you to understand all the possible ways to tell fmincon what your problem is. And also there are a lot of parameters to be set, so you can have different performance or different settings. There are small tricks here and there. Anyway, let's start from scratch. What I would like to do actually is going to be the longest one. This one, okay? So I'm going to use this many inputs, but I'm not going to use every one of them. Fun is going to be additional function outside of this main code. So you will be type in the name of that function at that file. So whenever fmincon reads this, it will go to that file outside of the main code to run what's inside for your function value. Okay? x0 is your initial guess. What is a and b? You can read what's going on here. A times b smaller than a times x smaller than b. Basically, this is similar to linear programming, but over here it's a smaller or equals to sign. Okay, so basically a bunch of linear equations. A can be a matrix, B will be a vector to indicate numbers of linear equations with smaller or equals to sign. How about AEQ, BEQ? Basically, EQ represent equality constraints. So AEQ and BEQ, this is basically the exact formulation for linear programming. Okay. LB, UB, lower bound and upper bound. So a vector of your variables could be bounded by a vector of lower bounds and a vector of upper bounds. They need to be written in vectors as well. Non-linear constraint. Non-linear constraint. This is basically another file written outside of the main code where you specify all of your linear constraints. And usually you will specify the constraints in this way. C, the return value of C gives inequality value. The return value of CEQ returns equality value. Okay, I will go through how to do it, but you need to understand what are those. Options, basically whatever you're setting will be defined in options. Okay, so let's work on this. Now, let's try to solve a nonlinear problem. For example, we're going to minimize x1 squared plus 10 times x2 squared, and then subject to, for example, x1 has to be within 0 and all the x must be within 0 and 10. And also, for example, I have another ellipse. So maybe x1 square about 3 times x1 square plus about 5 times x2 square must be smaller than some value. If I want the radius to be 6, then 36, maybe something like 50. Okay, it's gonna be an ellipse, and I want my solution to be inside this ellipse. Actually, yeah, that's fine. Maybe another one, maybe one equality constraint. So let me make a horizontal cut. So or a linear line. Then maybe x2 equals to 0.5 times x1, maybe plus 4. That's an equality constraint. Okay, so the solution is gonna be 0 and 4. To make it more interesting, maybe another one. Okay. Let me make another one. X1 times x2 must be greater than some value. Maybe 4. Something like that. Okay. So it's gonna be a curve like this. So the answer would be this plug in this. Then that's the answer. Anyway, a bunch of equations. Now, you of course need your initial gas. So in my case, you don't really need to specify all of your variables in this way. It's just telling you input 2 is this, input 3 is this, okay? But you can name your variables any way you want. For myself, I'm gonna call this, I'm gonna say, do I need iteration? No, I don't need iteration. I need, okay, just x0, yeah. But I like, in this case, I will use capital X. It looks nicer. So for myself, I'm gonna just say, or just capital X, and then it's gonna be the some initial gas. So let's say I have 7 and 8. Okay, it's my initial gas. Okay, now the lower bound and upper bound, I'm gonna say lower bound is going to be 0 and 0, upper is going to be 10 and 10, because it was described here, the lower and upper bounds. Okay, if you are using horizontal variables, treat them as horizontal ones consistently. Otherwise, it will be too confusing for a computer. Now, we need to save this file. I will just call this a test 001. And then I need new files. I'm creating new files. Over here, you're going to just write this file as a function, and you're going to return values. For example, y, and you are going to name this function any way you want. I am gonna say my func, and you can only use one input. For example, x. Okay, but when you call this function, what you throw into the function is actually a vector. So the name the input, only one input, but it's actually a vector later on. Okay? So I'm gonna copy this portion where I wrote my objective function. Here you are going to physically write down how you evaluate the function. So basically y equals to the first input square plus 10 times the second input. That's it. That's how you describe the objective function. So whenever fmincon needs to evaluate what's the function value of certain variable, it will put the variable values into this function, and the m file right here is going to evaluate the function value for fmincon. In return, fmincon receives the function value of that particular variable. Okay, then save the file. Of course, you have to save the file as the name of, in my case, my func. Okay. I already have one, so don't screw up what I did in the past. Let me call this my func1. Okay. Okay. So since I call this my func1, later on, I will need my func1 right here as the first variable of fmincon. Let me just write down fmincon here. Fmincon. Actually, what you can do is to copy the whole thing, so you don't need to memorize it. Okay, so the first one is going to be the name of the objective function. Then your input, your initial guess. I don't plan to have linear functions, even though I have some, but I'm not planning to do so. I'm planning to describe them in a second file, a third file. So what do you do for those four? You can just put empty brackets here. Just give fmincon nothing. Okay? Then my lower bounds, upper bounds. Okay? For nonlinear constraints, I'm gonna call it my con1. And then later on, I'm going to set up some options. Okay, so that's how you're going to do. And in return, the result could be in return, fmincon returns the optimal solution. So I'm going to save it as the second row of my X. Okay, so now I need another file. Create a new file, say func. This time, since you need to specify inequality and equality constraints, so you're going to say C and CEQ, and the name of the file my con1, and only one input X. So similarly, when fmincon needs to evaluate the constraint values, it's gonna be a vector of some guess from fmincon. That value will be sent to this file, and what you write here is going to calculate what's necessary for your constraint values, then return back to fmincon. So in our case, I need to describe these three constraints. So I'm pasting them here just to remind myself what those are. So in this case, I'm gonna describe my equality constraint first, because I only have one. So my equality constraint will be 0.5 times x1 plus 4 minus x2. Okay, so basically I move x2 to the right hand side of my equation. And that's basically how you evaluate. I need this to equals to 0. Okay. Next, oh sorry, since the value comes in is going to be given under the name of x, so this is gonna be x bracket 1 and x bracket 2. The first component of x, second component of x. Okay, next, now we have two inequality constraints. What you're going to do is say C1, basically the function value of the first inequality constraint. Okay, and here 3 times x1 squared plus 5 times x2 squared. Here I'm gonna minus 50. I'm moving 50 to the left hand side because I need the function to be smaller or equals to 0. From the description, it tells you it has to be smaller than or equals to 0. That's why this 50 moves to the left. Now my C2 is gonna be 4 minus x1 times x2. Because I'm moving the left hand side to the right, keeping the what you have on the right as your constraint, and then this gonna be smaller than or equals to 0. That's it, save. Okay, what do you do next? Run the code. I forgot about options, so it will give me errors. So let's learn how to do options. What you can do here is to find options inside this description of fmincon. Whenever you see options and there's a link for you to click, you can click in, and it tells you, oh, this is how you describe options. Actually, there is something called, let me see, I couldn't find it. Set, not optim set. Optim set, I think. Yes, optim set. Optim set. Optim, or you can directly search for optim set. Yeah, optim set. Options equals to optim set. So here, options equals to optim set, and you need to specify what you are going to set. So you can go back here. You can choose. Actually, you can choose the algorithm. So you can say algorithm. I'm gonna use SQP to solve it. Okay, it tells you can use SQP, and there are some other methods: interior point, active set, trust region. So it's like there are so many methods to be chosen, and we only cover SQP, but we don't even cover the complete procedure of SQP. We only derive QP up to a certain point, where there are 2 more equations they are not yet linear, so that couldn't be solved by linear programming, and we stopped because QP cannot be done by what I described. It needs further procedures, and there are researchers who develop different strategies to do that. And when you are able to solve QP, if you solve it sequentially, then it's SQP. Okay, but MATLAB already has the whole procedure, and MATLAB will automatically adjust what needs to be done for SQP. You don't need to worry about it. However, you cannot figure out how MATLAB did it. That's the issue. So sometimes when you try to publish a paper, if you say I use fmincon, that's okay. But the main contribution of your paper has to be something else. Maybe a thermal system or some mechanical design. So fmincon becomes a tool in your in your paper. That would be fine. However, if you are truly focusing on optimization, you try to publish in some topic related to optimization, and you use a commercial package, then the reviewer usually will challenge you. Hey, what's your main contribution? If fmincon did the most of the job for you, what did you do? Okay. So it really depends on how you do your research and how you contribute in your paper. Okay, for myself, I usually just use fmincon because my main contribution is not to develop a new solver. My main contribution is to solve some unique problems, and I develop methods to formulate the problems, and that problem can be solved by any solver. It doesn't matter to me. My job was not to create new solvers. In that case, I can use fmincon, and that's okay for me. Okay, so it's up to you to how you are going to use it. So next, I'm going to turn on something called display. By default, it's off, so it doesn't show the procedure of iteration. So usually I would like to turn on display. Just say, I think it's iter. When you say iter, that means show the iteration right here, iter. Actually, there's more. There's a iter, detail, and so on. Just iter is fine. And let me see if I want to do anything else. I think that's enough. That's what I'm planning to do. So what's next? Run the code. The typo, display. Now run the code. Okay. Forget about, I should use bracket. Typo. Okay. Now do it. Done. Finished. Converged. What's the solution? Solution is 1.339 2.9874. But what happened so fast? It happened so fast. Is there a good way to realize what happened? Okay, let's read the iteration procedure. Initially, 0 iteration. It made 3 function calls. Okay, I will tell you why. It's trying to evaluate the gradient. And then feasibility means the objective function value. For optimality, let me see. None of the step basically is like an error. But first order optimality, I'm not sure what. Sorry, I was wrong. This is feasibility, this is optimality. Okay. So optimality is function value, objective function value. Feasibility is basically the constraint value. This has to be. So the solution was not feasible? Hold on. Let's plot. So sometimes even when I'm reading this, I'm not even sure. This is the f value. This is the objective function value. This probably is the gradient, because first order. So it's like a gradient f. But feasibility should be the value of your constraints. However, it doesn't go to 0. It tells you it converges to an infeasible point. So it didn't converge well. Maybe SQP couldn't complete it. Let's try a different method. Let's just try different method just to see whether fmincon can find the true solution. So I did another one. It took me 237 iterations and converge at an infeasible point. What's the solution? It's different. So different algorithm gives you different solutions. Let's try active set. Did I make something really hard for fmincon? Let me try a different method. Okay. Active set. Also infeasible. Okay, I need to plot the function to figure out what happened. Now, how do I make plots? So what I'm gonna do is to make the plots in the main code. So over here, I'm going to define my plotting zone. It's going to be inside 0, 10, and 0, 10. And now I'm going to plot the objective function. So figure 1, easy contour, and my objective function looks like this. Actually, I can copy this. So you need to decide how you plan to provide fmincon. Maybe symbolic. So you can directly do this. And then plotting inside zone. Okay? Now hold on. And I want to change the thickness of the contour. So object find line width by default is 0.5. Save them as H, a handle. No, not object find. Find object. Then set. Based on what we did in the past, the second one is the object for the contour. First one represents the box of the figure. Second one, and then change the line width to 2. Okay, let's run this just to see if we did it right. Contour typo, easy contour. Okay, nicely done. Convert the contour to dash line, so line style. And then try this, and also axes equal set GCF color white, changing the background to white. GCF means handle of current figure. Set GCA, handle of current axis. Font size 15. Do it. Okay, maybe 20. Larger fonts. Okay, very nice. So that's how the function look like. Maybe set the figure larger. GCF. Location or position? Position. 100, 100, 600, 600. Okay. So that's the objective function. The minimum point is right here. Now, I'm going to plot the constraints. So the first one, easy plot this function. Let's see. Oh, it's a like an ellipse right here. Let me just give another handle h1. Okay, so set h1. What's the warning? Function plot. Okay, let me try this. I never tried this. A new function. Ah. Function plot. I don't like function plot. I like easy plot more. MATLAB asked me to do this function plot, but it doesn't has the same functionality of easy plot. Okay, now I want the line color to be red, and then line width to be 2. Okay, now second function, h2, copy the whole thing. Second function, let me do this guy. Here I'm gonna just say minus x2. That's what I did in the constraint. Set h2. Let me change the color. Let me use blue. Okay. Ah, this is why. The second one is a equality constraint. You must find solution on the blue line. However, your red ellipse only allows what's inside, underneath the red ellipse. So there's no overlapping. That's why fmincon couldn't find a solution. So let me change this to 100. So my constraint right here changed to 100. This one changed to 100. So I will have a larger red ellipse. And the plotting changed to 100. Okay? So actually the feasible space is right here, so small. Maybe even more, 200. Okay, now the feasible space is actually here. And we have a third function. So sometimes plotting out is nice. You can see what happened. But if it's a 4-dimensional problem, you can even make a plot. 3-dimensional problem, you can try it using isosurface. But above 3-dimensional, it's impossible, almost impossible to make plots. Now the last one is right here. So 4 minus x1 times x2. Yeah, and this one, I'm making it green. MATLAB green is too bright. If you don't like this bright green, you can specify green color by yourself. You can try 0, 1, and 1. That's exact green you had. If you put a little red and blue in there, it will be darker green, supposedly. RG. How come it's still the same? Is it running in the 255? No. 0.8? no, it become bright. So how about this? I'm tuning the. No, definitely not this. 0.2, 0.2, and 0.6, I guess. Yeah, see, it's a darker green. So anyway, it's a if you, this is basically R, G, and B, red, green, and blue, and if you have 100% of green, by default it's a really light green. 0 is like black. So if you make the number lower, then it's a darker green. However, you need a little red and blue to make it more like the green you like. Anyway, this is this portion of the blue line is the feasible space, because you need this red area underneath. Actually, you need this this area above the green curve as well. So only this portion, small portion, from the green edge to the red edge on top of the blue line, that's your feasible space. And by human observation, this point is the optimal, because that's where you have the lowest function value. Okay, so it should be somewhere close to 1 and 4.5. So let's see if fmincon give us the result. Almost close to what I told you, 1 and 4.5. Okay, so that's the solution. So this is by active set. And what's interesting about active set is right here it tells you it was an infeasible starting point, and over here it tells you it did use Bayesian. It used Bayesian. Okay, Bayesian modified twice. But the problem is you don't know what MATLAB did. It just give you some information. Okay, let's try a different method, SQP. Run the code again. Okay, so SQP just show a bunch of numbers. Okay, so let me show you another trick to figure out what MATLAB did. Okay, so over here, whenever I'm going to make breaking points inside my functions. So whenever I run fmincon, if fmincon runs my additional functions, it will stop there. Then I realize, okay, this is where you come in and evaluate the function value. So let's run the code. Okay, so this is the first time fmincon goes to the extra files. So what's the value? 7 and 8, basically my initial guess. Okay, let's continue to go. Later on, it goes to my constraints. It's trying to evaluate the constraints, and the values are 7 and 8, the initial guess. What will be the next? The next time it goes to the function, it's not 7 and 8 anymore. It's something close to 7 and 8. But how come it looks like 7 and 8, but maybe it's not? So let's see. Let me multiply. We will see later on. I will tell you how we can observe this. Let's continue. So for the constraint, it went to the same place, 7.00. You have no idea what it is. Okay. A third time, also same place. Fourth time, okay, now it's going to somewhere weird. 2.7 and 5.39. I bet now is in the second iteration already. Based on what MATLAB did, some evaluation close to 7 and 8, it moved to 2.78 and 5.39. So this was done by the algorithm MATLAB used. Okay, but just manually reading those numbers doesn't help me. How about we plot those positions whenever MATLAB comes into the functions? Then we will know. So from this observation, you already know the same set of variables will be sent to my objective function and also my constraint. So if I want to do plotting, I don't need to write plotting codes in both sides. Then if you do that, you are basically plotting the same thing twice. So you can choose either side to make some plots. Let me make some plotting codes in the objective function. So here I'm gonna say, whenever you come in here, I'm gonna plot a point of that position. So just say plot x1 and x2. But whenever you plot, please plot as a small circle in the color of black and marker size 8, line width 1.5. Let's do that. Okay? So if I do that, I don't even need to pause. Just run the code. You do, how come? Oh, B represent blue, sorry. Okay, if it's blue, it's blue. So it seems like it's moving toward this way. But if you zoom in, this is where your initial guess is. Keep on zooming in until you realize the trick of MATLAB. Do you know what this is? Finite difference. This is your initial guess. So MATLAB made a tiny increment along x direction and made another tiny increment along y direction. This is exactly what I taught you. So what MATLAB was trying to do was to evaluate the gradient. Okay? So if you look outside of the code, how come there's 3 types of function calls? Those are the 3s. A point, a movement toward x, a tiny increment toward y, and then 3 evaluations. So in the first iteration, MATLAB evaluate function value 3 times. And those are the 3 positions MATLAB went to. Now, let's check the I think this is the second point. So after reaching to the second point, did MATLAB evaluate the gradient again? Yes, it did. So again, the gradient was evaluated at this point. Okay, so I guess third point is this one. Same. Always use finite difference. Let's check when it's closer to the optimal. Okay, so there were 2 evaluations. Okay, let's check this one first. That's, I think, the fourth one. Again, finite difference. Okay, last one. Let's check the last one. Actually, that's 2 more. Okay, so you can tell 3 points here, 3 points here. You can check again. Yeah, 3 points. Check this one. This is the final. The last position fmincon found. So whenever it went to a new place, it will perform finite difference to check the gradient. So that's what this algorithm did. Let's check what algorithm we used. We used we used SQP. So basically, SQP didn't even try to evaluate a second order derivative. Okay, because when we introduce SQP, remember we try to reduce it to linear programming. But inside SQP, we do sometimes need to compute gradient. Let's try different methods. This is a method called interior point. Okay. So this method looks similar, and and from here you can tell every time 3 evaluations. So definitely it's testing the gradient as well. So nothing interesting to check. So let's check another method. I remember there's a third method, active set. Let's try active set. Active set. So from here, it's always using gradient, but it mentioned about Hessian modifier. I guess it's a approximated gradient, approximated Hessian. But anyway, let me just quickly check. I believe whatever it's doing is gradient finite difference. Yes, always the same. It's doing finite difference. Is there a method that performs second order derivative? Let me try this truss region reflective. Okay, it doesn't. Okay, if it doesn't solve, it doesn't solve. Okay, it will just give you error message. So it tells me it doesn't solve the problem. How about this SQP legacy? Okay, this doesn't solve as well. Okay, upset doesn't support. Oh, I need to use a different method to set algorithm. Okay, then let's try it. Optim options. For some reason, you have to use optim options for this method. Optim options, and then I guess you can do this. I guess the rest is the same. So let me hide this line. No, opting options. Oh, typo. Optim options. Well, okay. So this is a different approach. So I'm not planning to figure out how to do it. So let me just get rid of this. Anyway, it seems like in all fmincon methods, besides the SQP legacy, they all figure out the gradient. Okay, I'm not sure if if any method computes Hessian. Oh, about the output. Let me see. Including gradient and Hessian. It just tells you what's the Hessian. Oh, Hessian function. By default, fmincon approximate the Hessian using finite difference, or use a Hessian multiply function. Okay. Yeah. So that's why it didn't try to compute the second order derivative. Okay. Now, I haven't told you about the outputs. What can you get for the output of your fmincon code? So what I usually get is the value of your new solution. Sometimes you can also get the function value. So you can say fval. Okay, this is what you usually need at the end. What's the final function value? So fval. This is the solution at the function value at the optimal solution. Okay, so you don't need to compute by yourself again if you type in this way. Okay, so from this example, I believe you can already do a lot in your project. Okay, I think if you get to know how to do this, you can already do a lot in your own research. Because let me show you one of my PhD work. Let me see. I'm not sure if it runs. Let's try it. What is it? It doesn't run. Okay, because in MATLAB version changes, so sometimes you have some old codes. It's written in the old functions, and in new versions you need to rewrite, so it actually doesn't run. Let me see if I have. How about this one? There's a button called email results. Why do I have a button of email results? I turn on multiple computers, run the code. When the code is done, send an email to me. So I can wait at home, That's what I did in the past. Monte Carlo, okay. It's I'm not sure if plotting. Don't do Monte Carlo. Okay. It is running. Okay, no, it's not. Some there are codes written more than 10 years ago, so it doesn't run in this version. But anyway, you can see in my PhD work, I basically plot things in exact same way, and if you go in to read some of my codes, I will show you. Maybe solver, not solver. Somewhere routine main optimization, maybe. fmincon here. See? So in my PhD work, basically I formulate different things, and at the end, fmincon. Okay, but it's a bunch of what I was doing. If you are interested, let me show you what I did. This one. So I was trying to solve some problem called optimization with uncertainty. Hold on, yeah, it was plotting. Yeah. So what happened was, if you this is a very simple linear optimization problem, constraint optimization. Everything is linear. However, traditionally, if you have a design point here, and since the gradient, the descent direction goes toward left, so that would be your optimal solution, 24 and 96. However, if there's uncertainty, when people say there's uncertainty in a variable, that means that variable changes with some standard deviation. And we usually assume it's a normal distribution. So when we plot some kind of ellipse in the feasible space, a design space, that means that range represents certain percentage of the position. Okay, so I was doing this optimization with uncertainty, and that's a big issue in the reality, because traditional optimization doesn't consider uncertainty. So if you put your solution in the reality, you will have a good chance of failure. So if we look at the optimal solution that we got earlier, there is a 50% of chance that your function value, constraint 1, will fail, because there's a 50% of chances your design point is above there, on the top, in the pink area. And there's another 50% of chances your third constraint will fail. In total, your failure rate is 90%. So optimization is not trustable if there's uncertainty. But in my PhD, what we did was we tried to formulate new constraints, more conservative constraints. And they are constraints like offsets coming into the design space, feasible space. They're offsets. And if you can formulate those and try to find solution over there, you can see your optimal will, the distribution, the range will have like a tangent to the original constraint. So what you get at the end is you can control the failure rate. If you say the failure rate, this is basically failure rate. If the failure rate of your G1 has to be within 0.13%, you can precisely control the failure rate right here, because the outer contour was representing 99.87%. So what's outside is 0.13%. Okay? So, but for G3, if you set up the failure rate to be 2%, then the smaller ellipse is having tangent with G3. So you can be sure the failure rate is 2%. So nowadays, this is called reliability-based design optimization. Okay, this is what I did in PhD. And there are already this kind of optimization being done in commercial products. Okay, so manufacturers are controlling the reliability of their parts, so their parts will die exactly when your warranty finishes. For example, how come you change your cell phone when the warranty reaches to the end? Because many parts or some critical parts inside your cell phone will die. They control the reliability so precisely. So even your warranty finishes, you cannot continue to use it unless you change some important parts. Okay, so now, do you remember 10 years ago, like some famous cell phone which survive 20 years without dying. A055. No. What's the name? The phone that doesn't die. What's the number? Oh yeah. What's the brand? Ericsson? Nokia series. Nokia series. They all survive much longer than the warranty. Why? Because at the time, reliability and engineering was not that good yet. So people didn't know we can control the reliability of every part inside your product. So 10 years ago, whenever we sell products, if we sell poor products, usually your product don't sell. So usually people try to maximize the performance of their phone or their products. So usually people overly design the parts. Either it's too strong or performing too well. So they actually they make less money. Because if you want to make your product stronger or performing better, you need to spend more. The budget is increasing. Nowadays, since people can precisely control the reliability, they can reduce the performance and reduce the budget simultaneously to find a balance between performance and budget and cost. So at first, when you get your product, it's perfect, but it dies with time until it reaches the end of the warranty. Some part inside will die, or some part inside will performing so poorly you want to make a second purchase. That's the trick nowadays. Okay, so that's what I did. So in my research, how did I found those those new modified constraints inside the feasible space? It was based on certain math. So my main focus of research was to derive some constraints. I found some derivations mathematically, and I use fmincon to formulate those modified constraints. So let me show you what's the end. So at the end, let me see. Yeah. So at the end, actually, this is you couldn't understand what this is about. But did I show the final? I have a simple original objective function needs to be rewritten in a certain way, like this, so I can get that offset from the original constraint to somewhere inside the feasible space. So it's purely math. This derivation was my main contribution. My main contribution was not to find a better solver. That's why in this particular research, I can use fmincon. Okay? I don't even need to tell people I use fmincon, because it's not the main contribution. Okay? So let me show you the paper I wrote. 2009. 2009, how old was I? 28 years old. Who is older than 28? Not yet, right? Oh, you look like 23. Okay, so yeah, this was my first conference paper that I think is a good one. And this one was converted to a journal paper. So if you want to take a look, this is a journal version. Okay. So it's about distributions. It's about math. Those are the math that I did, and those figures were done by MATLAB. As you can see, the way I plot figures didn't improve since 28 years old. So yeah, a bunch of math, and yeah, see, exactly like what I'm showing you in the lecture. But the reason why I'm showing you this is just to tell you whether I'm telling people I'm using fmincon or not. So I just Google search and say fmincon. Just search the file. I never mentioned fmincon. Did I mention MATLAB? I never. So you don't need to tell people you are using MATLAB, because this exact research can be implemented in Python and can be solved by any Python optimization solver. It still publishes. So the solver or platform you chose were just tools. So when you do research and you are doing research related to optimization, you need to be aware of this. Usually, optimization is just a tool. But if you didn't learn this course, and if you just found some optimization solver online and just implement it and just use it, sometimes you didn't know what happened inside. Sometimes you can make mistakes, just like what I did earlier when I didn't choose a appropriate constant. So see, at the beginning, the optimization solver told me we couldn't find the feasible solution. So if you are just a regular optimization user and you use some tool to find solutions, that could happen. The solver can tell you, hey, I couldn't find a feasible solution. Then since you're the user, you need to figure out. So I think it's important to understand optimization and also understand the details inside the problem. If you give any solver a poorly defined problem, you may end up with poor results or infeasible results. Another common mistake students can make is when you use a solver and get solutions, and you just believe it. But sometimes maybe there's a mistake inside you didn't know. The solver eventually give you some solution, and you thought you didn't make any mistakes, and that's where that could cause a problems at some period of time. So please make sure what you are doing exactly. Okay, otherwise you could make mistakes anywhere during your research. Okay, anyway, let's get back to the second part of my lecture. I want to talk about GA. So GA is very easy and can be easily implemented. But I do need to take some notes during my explanation. Okay. So, teaching optimization. We already covered fmincon. Today is May 22nd. Let's talk about genetic algorithm. Genetic algorithm. GA. So what happened was there's something called chromosome. In Chinese, it's 染色体. If you don't know what is chromosome, okay? Because many of you may not be from the biomedical field, but if you're foreigners and you cannot read Chinese and you don't know what is chromosome, well, in DNA you have something like this, and you have stuff inside. I'm not from the biomedical field. I don't know what's the proper term of this. So for our DNA, it's actually a long string of codes. So basically, GA implement that idea to by making long strings of information. So in GA, what they like to do is to have a long string of numbers, and usually 0 and 1s. It could be in any length based on your problem. Okay, so what do people usually do? They will convert everything to 0 and 1s. So for example, I have in the problem I showed earlier, let's say I have x equals to x1 and x2. And let's say x1 and x2 ranges from 0 to, how about 32? No, not 32, 31. Why 31? I will let you know. Because if I use 4 bits, 0, 0, 0, that is 0. Binary codes. 0, 0, 0, 1, that's 1. Da, 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 da. Oh, by the way, maybe I write more. Binary. 0, 0, 1, 0, that's 2. 0, 0, 1, 1, that's 3. Sorry. 1, 1, 1, 1, that's 31. Is it? Because 1 plus 2 plus 4 plus, oh no, that's not 31. So I need one more bits and one more bit. So plus 16. Is it? This is 12. This is 3, 15, 31. Yes. Delete. Okay. So 5 bits. So if that's the case, if x1 and x2 they are integers, then I just need 5 bits to represent x1 and another 5 bit to represent x2. So at the end I have a long string of 10 bits. And this portion represents x1, this portion represents x2. That's my chromosome. So in reality, you have a design space x1 and x2, and this is 0, 0. The largest value you can get is 31, 31, right? This is 0, 0. So in total, you actually have 32 times 32 combinations. Combine. Is it? right? Combinations. If you try all of them, that's 32 times 32, right? So how many combinations you have right here on the left? 2 to the power of 10. So it's 1024 combinations. Should be the same as this one, right? This is basically, is it? Yes. Exactly. So this 10 bits of 0 or 1 has 1024 combinations. Each one of this string represents a point in the design space. So if one of these 1024 points is a solution, let's say you have a function inside the space, x1 and x2 follows this rule. They are integers, they ranges from 0 to 31. What you can do is to try to evaluate the functions 1024 times and then pick up the best one. That's your solution. But maybe it's a stupid way to try all of them. Try all 1024 times. It's very stupid. However, can you solve this by a continuous fashion? What do you mean by continuous fashion? Just treat it like continuous problems. However, this problem was integer. Everything was integer. So sometimes you cannot. Let's say x1 represent number of people. x2, let's say x1 represent number of boys, x2 represent number of girls. You cannot have 1.5 boys. You cannot cut a person in half, right? So maybe the objective function is some factor in terms of with respect to number of boys and numbers of girls. So for that kind of factor, you cannot take derivative because you cannot use finite difference. You cannot just cut away one person's hand and say, let's see how the objective function is going to change. It's not going to change. Someone is going to the hospital. So finite difference doesn't work in discrete problems. You cannot take derivatives. So one way to handle this kind of problem is to use the stupidest way. Try all of them. A second way is GA, some method like GA. So what GA did was, first we need to know some terminology. They have a name called generation and population. Okay? Well, what does that mean? Generation is just like an iteration. But since they are following genetic theory, they want to use the terms that fits the story. So generation is like our generation, next generation, a third generation. Our grandparents is the first generation. Second generation is our parents. We are the third generation, something like that. And in optimization, it's like our initial guess would be our grandparents. And then the second iteration, we came up with our parents. In the third iteration, us was born, something like that. So GA deal with those numbers and then treat those numbers as chromosomes and try to give birth to new child. And if the child perform poorly, they kill the child. I'm not kidding. They only keep the nice kid. They only keep the good kid, kill the rest of them. So, why do they do that? Because in genetic theory, researchers believe during generations, all kinds of species, they mutate. So whenever there's mutation, new kinds of species was born. Sometimes some species can survive the disaster or whatever environment in the world. Some others cannot. So in genetic theory, they believe whoever survives or whoever can fit the environment stays alive. Okay? GA is using the same concept, but GA did it numerically or systematically. I'm gonna tell you how they did it. Okay, so generation basically is the iteration. But how about population? Population is like a number of random gases in each iteration. So it's like you can say your population is 10. That means in the design space, you randomly make 10 guesses. Okay? It's just like that. Okay? Now, second, what they usually need to do is to they usually need to do some kind of feasibility check. Okay? Because maybe in the design space, if you have a constraint function, for example, like this, and here's like a G1, and here could be a G2, like this. For example, this is G1. For example, this is G2. And for example, your design contour goes this way. The objective function, F descent direction. By observation, the intersection point of G1 and G2 should be your solution, right? But now there's no method can tell you that's the solution, because everything is discrete. You cannot take derivative. So now in first generation, we just randomly generate 10 points. So basically, there will be 10 random gases being chosen, being created inside the space. Some of them will be inside the infeasible space. So what you usually do is you need to basically check feasibility of each chromosome. Chromosome. Basically, each random design. And GA can be done so differently from each designer. You can throw away the infeasible ones. So, for example, your population equals to 10. Okay, so the bracket red color represents some example. And when you check, for example, there's 3, they are infeasible. So what you can do is to basically throw away, kill them. And then randomly make or randomly generate 3 more. And of course, you need to check those 3 more until you have 10 feasible points. You can keep on doing this until you get 10 feasible ones. Because usually in GA, you don't want your population that has infeasible points. You want to kill the bad kid. Keep the good kids. Okay? If a kid doesn't have arms, kill it, bang. If a kid has no eyes, kill. You need someone looks like a person, okay? Something like that. Okay. Once you have 10 feasible points or 10 feasible chromosomes in your population, what you can do next is to check the fitness value. This is the key of GA. What is fitness value? Something related to your objective function. So usually we give a score to the chromosome. The scoring system could be ranging from 0 to 100, 0 to 1. You can define your own score. So you need to specify what is a good score. If it's reaching a really good objective function value, give a good score. So it's like a scoring system. And sometimes you don't define this scoring system well enough, so your GA doesn't converge, or your GA is not effective enough. So usually if you write a paper, you need to specify how you formulate your fitness value so people can follow you. Okay? And usually if you are minimizing, for example, you are maximizing your objective function value, then your fitness value could be your objective function value. So larger the better. So fitness value could equals to your objective function value. Okay? But it really depends on how the problem is. Next, something called selection. So we usually want the chromosomes with better fitness value to have better chances to be selected. And we want the minorities to be the ones with lower fitness values because we don't like them. They are poorly performing kids. They are feasible points, but they're poor. So we don't want them to be chosen. We give them a lower chances to be chosen. Okay, so in this selection trick, there's a famous one called I forget the English name. Roulette wheel. There's a roulette wheel selection. What does that mean? It's like you are creating a wheel, and then you are cutting the wheel into smaller pies, but they have different sizes, and you will put, for example, chromosome 1 here if it's having the greatest fitness value. Let me just say fit for short. And for example, there's another one, maybe C6. Maybe it's having the lowest fitness value. So it's like you are creating this roulette wheel, and you are giving them certain percentage, but the percentage is a function, the area is a function of how well that chromosome is. This is a but you know, in computer codes, you don't really build a wheel. You just write down the coding to implement this concept. So it's like okay, once you have a roulette wheel, you basically like throw a how do you say call that? What's the name of this? What's the name of this? Like throwing something to hit the game. Dart. Yeah. Like throwing a dart, and then you have this roulette wheel on the wall, and it's like throwing, and then once a specific chromosome was chosen, that one will be moved to not like a next level. So before selection, we have 10 chromosomes. At the end, we want to choose 10 out of the pool. So if the pool has 10, at the end of the selection, we also need 10. But the ones with larger area, they have better chances to be chosen twice. So it's like we're making duplicates. We're making duplicates of good kids. Okay? So at the end, usually you will get same amount of chromosomes but the ones with good fitness value could be chosen multiple times. It's fine for this procedure. So it's like whoever can survive better can have more duplicates. We want more of those good ones. We want less of those poor ones. But we are not that lucky. Usually when we do this, none of them is the optimal solution, right? We just, because at the beginning we just randomly generate them, we evaluate how good they are, and then we start to duplicate the good ones, kill some of the bad ones. That doesn't mean any one of those are the good ones. So what's interesting about GA is next, something called crossover and something called mutation. Those could be done in different sequence. And the way you treat crossover and mutation can be done differently. But the idea is like this. For crossover, you will pick up 2 chromosomes. So for example, this is chromosome A, chromosome B, and then you are going to use a random generator to decide where does A and B cross over. Crossover means like making out and share their body parts. In GA, they exchange their body parts. So what does changing mean? So what was done is over here you have a random generator, random number. And if the random number is, for example, smaller than certain value, for example, smaller than, let's say, 0.3. If that happens, then let's perform crossover over there, over at this position. Or if that happens right here, so in this particular, at this particular digit, that's exchange our body part. So at the end, they will perform some kind of body exchanging. This is the, let me make a copy of these. if they, it turns out they are exchanging their body. It's like what they did at the end is, let's change our hands. Let's change our arms. My arm goes to your body, your arm goes to my body. Since we are all good kids, we are all performing pretty well, maybe when we exchange our body parts, some of your good genes goes to mine and I could become even better. The idea is like this. So crossover randomly decide whether two chromosomes need to change their parts. And sometimes if the random generating number doesn't go doesn't go below 0.3, crossover doesn't happen. So usually this crossover, we call this crossover rate. And when you publish a paper, you need to tell what's your crossover rate, what's the rule of your crossover. Okay? How about mutation? Mutation performs on individual chromosome. And similarly, you generate random numbers. And usually mutation rate is very low. For example, 0.05. For example, just 5% of chances mutation happens. And let's say you generate random numbers from the first digit until certain digit, and you realize, okay, this particular digit, when it moves to this digit, your random number happen to be lower than 0.05. What you will be doing is, if it's 0, change it to 1. If it's 1, change it to 0. So it's like randomly decide whether some of your body part change to something else. So for example, this is my body part. Okay, random generator said, you don't need this arm, you don't need your palm. This should be changed to something different. And you become a elite species. Like a mutant, right? Like a X-Men. Just mutate your body part randomly. Okay? Once everything was done, originally you have 10 populations. After the crossover and mutation, definitely you end up with 10 different stuff. 10 different chromosomes. There could be some of them remain the same, but many others become new species, mutants, or kids. Because when we exchange body, we say, okay, it's like we give birth to new child, right? So what you usually do at the end is to check the fitness again. And what you will do is to keep so-called the elite chromosome. The best ones. So for example, keep the best one. After you do that, what do you do? You will let me just do this. You keep the best one, right? After that, just keep the best, basically the elite, and move on to next generation. So basically, whoever was not chosen as the elite member were killed. And when you move on to the next generation, you will again randomly generate new chromosomes, and then repeat this process again and again. When does it converge? You never know. Usually we will set up, so we will repeat the procedure until max iteration number reaches. We usually don't know when it's gonna stop because the process is so random. You never know when you can find a solution in the feasible space. Okay? That's GA. Let's try this in our example that we showed earlier. Okay. So if you didn't get the note, it's okay. I will give you later on. But I need to move on back to MATLAB. Turn off some of my old codes. Get back to this one. Turning this off. I don't need these. Turn off. Okay. So to keep what I have right here, so I'm gonna save this as a different file name. I need to turn off this. Okay. Test 2. Okay, so now I'm trying to do this without using fmincon, so I can get rid of those, but I can keep the rest. I don't even need an initial guess anymore. So this time x1 and x2, they are integer. Actually, I don't need them to be integer. I will tell you what I will be doing. So let's see. I need to create chromosomes, right? For let's say pop equals to population, pop equals to 10, and iter basically is your it's your generation. So max iter. Also, I would have a while iter that is smaller than 100. I need to do iterations. So when actually I would say, yeah. So when we reaches 101, this while loop will finish. So 100 will be my max number of iteration. Now every time inside I need to generate chromosomes. So I would say for I equals to 1 to pop. Do I do this? No, I will use while. So I will say I equals to 1, and then while I need to generate a matrix. So my matrix, I'm gonna call this matrix X, or just chromosome, chromosome. And I'm gonna make it empty first. So while the size of chromosome, and I'm computing the number of rows, when it's smaller than pop, I need to repeat this while. So this while will be done repeatedly until the size of this chromosome reaches 10. Then we will be out of this loop. So whenever we're inside, I'm gonna generate one line of this random. So it's gonna be chrom. How do I? Let me see. I want to generate random 0 and 1. Let me just do this. Chrom, and then the ith one. So now this is where you define random numbers. So you can say rand 1, and then we need to define the size of it. So now I'm not going to use just 5 bits. It's not enough. I'm going to cut the design space into 10 bits for each variable. So I'm going to make 20 bits. So when I use rand, that means I generate 20 random numbers ranges from 0 and 1. If I do a rounding, so whatever that is smaller than 0.5 becomes 0, larger than 0.5 becomes 1. Okay, let's see if I can have my first generation of chromosomes. Sorry. I forgot to have sorry, I equals to I plus 1. Okay, so let's check my chromosome. I have a bunch of I have how many columns? And now I have 11 of them. So that means I don't. This is the first one. Second. How come the 11th one come in? Goes to the 10th one. Chromosome. I goes from 1, right? So the first one, then when it's 10, I don't need I don't need to create one more. So I can just I don't need the equal sign here. Then I will have chromosome 10 by 20. So I have 10 chromosomes. The for each row is basically random numbers, random 0 and 1s. The length is 20. For the first 10, I will treat it as my x1. Second 10, I treat it as my x2. Now I need some kind of converter. Whenever I have a binary code, I need to convert it back to x1 and x2. So I need to actually, whenever I generate one, I want to check if it's feasible. If not, I don't want it. I will. So first I need to generate it back to the decimal value, and also I need to check whether it's feasible or not. So what I'm going to do right here is I have a range from 0 to 10. I'm basically cutting it into how many pieces? 2 to the power of 10 pieces. So so here, let me call this X for each chromosome equals to my lower bound, the first one. lower bound, upper bound, the first one, minus the lower bound. This is basically the range. And divided by the number of cuts. So supposedly I should be having 10 to the power of 10th. So I'm dividing this into 2 to the power of 10. 2 to the power of 10. That's 1024, right? Yeah, 1024. I'm cutting this into 1024 pieces. So this range is being cut into 1024 pieces. Then I need to know which one of it. Oh, maybe I have this binary to decimal converter. Binary to decimal. Oh yeah. Yep, that makes my life easier. I just need a string. So multiplying which one, right? So binary to decimal, but I need a string of it. So it's gonna be number to string of this particular chromosome. But does it really provide what I have, what I want? Check it right here. So let's check the first line. So it will give me a bunch of let me see what does the answer look like. There are spaces in between. So I need to get rid of the space character. What I can do is to set difference answer and then the space. No, cannot do set difference. How do you get rid of this? How do you get rid of so text string exchange something, there's a command for this. Like a replacement, something string replace, let me find it. This one. This one. Replace text using regular expression. So regular expression replace. Okay. So I will be replacing. For example, 0, 1. Replacing this, and then I'm telling the MATLAB the space one will be replaced by empty stuff. Oh no, it's not like that. Let me see. String expression replace. Yeah, why not? Okay. I got it. Cannot be an empty one, but should be able to do this. Yes, perfect. So once you have number to a string, you need to do this. You need to have replacing a string, but replacing what? Replacing the where? This whole thing I will end up with a bunch of 0 and 1, but also a lot of spaces. The space character, replace it by empty character. Okay. So this will give me a decimal number, ranges from 0 to 1023. Yeah. So if it's the last one, 1023. So here I need to minus 1. Because this ranges from 0 to 1023. This has to be cut into 1023 pieces. 0, 1, so this is multiplying the 0. That's 1, oh no, still 1024. 1023. Cut into 1023 pieces. And you basically have 1023 from 0 to 1023. So you need to add the lower value here. Okay, so that should be my first value of X chromosome. Then I need to, I'm not sure if it's right, I need to check later, but copy the whole thing and check my second one, basically upper bound minus lower bound. Same thing here. Same chromosome. No, sorry. This should be the first 10 chromosome. This should be the second, the last 10 chromosome. 11th one to the last one, and here lower bound 2. Yep. So let's see what happened. So now my random number, I generate a number. Let's generate a bunch of them and see if I'm doing this right. Let's make a plot so I know if I'm right or not. For now, I need to check whether it's not, whether it's right or not. So just plotting those points. Small black circles, marker size 8. Okay. And let's make a lot of them. 10,000 of them, just to see if I'm making this random generator properly. Is it too many? Okay, don't wait. Don't wait. Oh, okay. I made maybe too many. Let me say draw now, just to make sure. And just 1,000 of them. Okay. Let's randomly generate 1,000 of them, just to see if I'm writing this code properly. So yeah, it's very random, and I want to make sure I'm not generating outside of the lower bound and upper bound. I think it should be correct. I can stop. I think it's correct. Just to be sure, if I do this, but this time I try 10 zeros. This should give me 0. Yeah. And let's try 10 ones. This should give me 10. Yep. So I think it was right. So I only need 10 within each generation, 10 populations in each generation, and I'm randomly generating points. I can hide this line because I don't need to plot anymore, but I'm not deleting the line because maybe later on I need it. So here I need to check whether the point is feasible or not. The way to check it is actually plugging in my func. Okay, plugging in my func. So this time I'm plugging in X chromosome. So it will give me constraint values and the equality ones and inequality ones, right? If it's a poor design, I will throw it away, right? So if because you don't know how many constraints and equality constraints are, so you need to say let me create a flag, infeasible flag. Originally it's 0, and you say if j equals to 1 to the length of c, not if, for loop, and then if cj that is larger than 0, then change the flag to 1. End. Right? End. You may not be familiar with this kind of coding. Let me formulate into multiple lines. So if any of the constraint value is larger than 0, it's not feasible. I will turn the flag into 1. And then I can check the same thing for equality constraint, EQ. But this time you cannot say equal to 0, because usually it's very difficult to get exact point on the equality constraint. You need to set up certain tolerance. Otherwise, you can never find a proper point. So I would say if this value, the absolute value that is greater than some small value, 10 to the power of, or just how about this, 0.01 as the tolerance. If that happens, make it infeasible. So if infeasible flag equals to 1, we don't want this particular chromosome. It's a bad chromosome. So I will kill this particular chromosome, make it empty. And then if it's not a bad chromosome, I will keep it. And I equals to I plus 1 moves on to the next chromosome. Okay? Let's see what happened. So in this case, I want to draw the ones that are good. I don't want to draw the poor ones. Let's see if we can get 10 of them. See, I found a bunch of feasible points on that design space only. I don't even know how many times GA tried, but I found 10 of them over here. If you want to know how many times GA tried, whenever you plug into the constraint function, you need to count it. So you can set up a function counter here. Function counter. You can set up as 0. So whenever you plug in, your function counter plus 1. Okay, and let's see how many times you need to randomly try so that you can find 10 feasible chromosomes. I bet hundreds of times. And you can see every trial is different. It's still trying really hard to find 10 of them. Okay, let's see. We already tried 23,000 times. So GA is not very good for this problem. It's so stupid. You try 23,000 times just for finding 10 feasible points on the line. It's not very effective. This problem is unique because we have such a small feasible space. This way of randomly generation, that random generation is really not effective for finding 10 points on the line. So how about this? We will turn off the equality constraint to make this process more effective. So how do you turn off? You basically just ignore this one. I'm just turning it off. This is turn off for GA, because otherwise it's not effective. So let's run the code again. Now you will see very effective generation, because it's very easy. Hold on, what happened? Sorry, even you turn it off, you need to say CEQ equals to empty, because you didn't return a value. You can return an empty value. So let's run the code again. Now you will see that line. I should turn off the line, blue line here. Okay, try again. So now we had 10 guesses, every one of them inside the feasible space. Okay, because but how many times we tried? We tried 47 times. Out of 47 random generation, we found 10 of them that is feasible. Good kids. Okay, now we can start to work on what do we do? We do a roulette wheel. We have 10 of them, but we are not going to keep the same 10 of them. If it's a good one, we are going to keep it. So when we plug in the earlier we check the this is for feasibility check, right? But we never check the performance. So now we already have 10 chromosomes. We want to check their performance. So I'm going to write a for loop from I cannot use I anymore. I need to use J. I use J already. Let's use K. Goes from 1 to the size of the chromosome 1. So for each chromosome, actually, yeah, that's okay. That's okay. When I have this X chromosome, actually I can keep a memory of those, so I don't need to do those again. In that case, whenever it's a good one, I want to save it. I want to save it as a value. So I'm gonna say chromosome and the ones you keep, and should be I of them. So that will equals to the random ones. Okay, earlier we randomly generate 0 and 1s and we convert into X by this conversion. And when we want to keep it, this is the one that is feasible, we will save it as a variable just to keep it. So at the end, the size of this X chromosome keep will be 10. We will be having 10 points. Basically, the coordinates of those 10 points. Now we want to plug into our function to see whether how good those 10 points are. So writing this for loop, I basically substitute them into this function. But this time plugging in each of them. So I will say y I not I k. So basically I plug into that function. This time I don't need to draw anymore. Okay. Let's just stop here to check whether we did it right. So we should have 10 values of y. Okay, they are different. And in this problem, we are minimizing. So the smallest, the better. Now you need to define your fitness. How do you, in this particular problem, you know, you have to know your problem. 0 is the best. And over here is the worst. 10, 10 is the worst. So you probably need to use those two function values as the lower bound and upper bound for fitness. So for me, I'm converting the function values into fitness value. Do I need to? Actually, I don't. Just use it as my fitness. So smaller the better, I guess. So wait, I need to convert them into 0 and 1 because I need, in the roller wheel, I need them to occupy certain percentage. So I still want to normalize those. So the way I'm gonna normalize them is to say y equals to y minus the mean value. Mean value is actually mean value is 0, but normalized by the largest value of inside this possible space. So basically substituting the function by 10, 10. 10, 10. So let's see. At the end, I should have a bunch of numbers ranging from 0 to 1, right? Okay. So, next I need to build this roulette selection. If you have a better fitness, I'm giving you better chances to be chosen. So I guess what I can do is I can sum them up. Sum of fitness equals to sum y. Because y equals to this, right? I sum them up. Those are the I'm gonna use 1 minus those. So when you do that, you have a bunch of greater numbers. Y is like because when it's lower, I want a better chances. So when I use 1 minus that value, so that means this 0.94, I want 0.94 to have better chances to be chosen. So now the sum y, sum of those values, or sum fit, the sum of those numbers are 8.14. So each of them has its own fitness over the sum chances being selected. So the select chance equals to each y over sum fit. So at the end, the select chances are those. Some has 10% of select chances being selected, others may have lower. Okay, now if you pick them up, it should be 100%. Right? So now you basically cut the 100% into small pieces. Some are greater, some are smaller. Okay. Now, what do we do? We need like a accumulate table. I need to accumulate them. So there's a way to accumulate. Accumulate. Let me think about it. Is it you? Really? Accumulate array? Let me see. Accumulate array. No, I remember there's a function that accumulates stuff. Really? Q sub? CUM as in accumulate sum. Yeah. Q sum. Okay. Q sum. Let me try it. Q sum. Q sum. For example, 3, 1, 2. Yes, Q sum. Okay, good. That's easier. So I need a accumulate array. So that would be my selection chance accumulate. So that would be Q sum of selection chance. Okay. So run it again. So at the end, each number represent it's like an accumulate percentage. The first one will have 9% being selected. Second one, when it shows 0.1964, that means a number ranging from 0.09 to 0.1964 should belongs to the second one. Okay. Now I can randomly generate 10 values ranging from 0 to 1, and no matter where it drops, that one will be pick up. Okay. So I need to now throw the dots, right? Throw the dots. Randomly generate 10 numbers. And I need to figure out which each dot belongs to which number. So what is a smart way to do it? I need to say, for example, 0.5 smaller than or equals to the selection chance accum. Then I will see 0, 0, 0, and 1, 1, 1. Okay, so darts smaller or equals to this guy. This gives me a vector with a bunch of zeros and a bunch of ones. So if I sum them, then I know how many ones are there. So for example, in this case, six ones that means it's in the range. Let me see what's in the range. I want to see the accumulate. I was testing 0.5, right? 0.5 was between the fourth one and the fifth one, so it should be the fifth one. So earlier, so that means 4 zeros plus 1. So this one, 10 minus this number plus 1. That would be the index for that particular chromosome. So the so dot particular dot. I need a for loop for ijkm m equals to 1 to 10 of them. Pop is, so this is actually pop. I'm not sure if you follows, but I'm sorry. Oh, this 10 should be pop, because later on if I change population number, this will needs to be changed accordingly. So this will be the index, the mth index. So I need to pick up the. Let me see. Let's try once. Okay, it doesn't work. What happened? Let's pause here. I should say dots mth. Run again. So the IDs are AA337947610610. Let's see why. Let's see the fitness values, the selection chances. So 8 was chosen twice because this is the 8th. It's having a greater number. 3 was chosen twice. Well, it was just random. Very random. Because the chances are so equal. They are so equal. Maybe it's because their fitness are so close to each other. Maybe I should use a lower, smaller value for normalization. So since I know the problem, I think if we plug in 0 and 7, use this function value as my limit, that probably will make a better performance. So 0 and 7 as the function value for my normalization, and try it again. So now we should be able to find, let's check the selection chances. Now you see different amount of chances of random points being selected. Some are greater. So 3, 4, 5, 6, 7. The 7th one has the highest chance. And let's see if the 7th one being chosen multiple times. Twice. 8 twice. Okay. 9th one once. So 1 was never chosen because the first one was poor. 4th one not chosen. 5th one was chosen. So 5th one was chosen 3 times. But he has a really low. Maybe it's just luck. Let me try once. Try one more time. We need to make sure this code is correct. Otherwise, we may be choosing inappropriately. Okay, ID and so here the greatest one is 7th one. Okay, we don't have. Oh no, this is 8th one. 8th one twice. Second one twice. I think the code was right. Okay, I think the code was right. Now, since we pick up some IDs, we need to now formulate the new chromosomes based on the IDs. So update chromosomes. Here I'm gonna call this a new, or say selected chromosome. Basically, it's gonna be the chromosomes with all these IDs. Okay, let me show you. So originally, these are the original chromosome. Maybe we will just, they are just 0 and 1s. These are the selected. So there must be some of them being selected multiple times, like this one. This one has been chosen multiple times. Because what is my ID? Number 4 was selected 4 times. What is the selection chances right now? The 4th one, well, it was luck. Okay, the 4th one was chosen 4 times. Okay, now we have completed selection. We need to do crossover. Okay, from the beginning, maybe I need to, this is for random generation, and over here there's a feasibility check, and here is for fitness check, fitness value check, and starting from here, this is for the roulette wheel selection. Now we are doing crossover. So for crossover, we need to pick up any 2. So for P equals to 1 to the number of population, for Q equals to 1 to the population number, from P. So P goes from 1 to 10, Q goes from P to, actually P plus 1. P plus 1 to 10. So that will give me any 2 combinations from 10 numbers. So it's like selecting any 2 numbers from 10. Okay. Then we are going to set up the so-called crossover rate. I'm going to set up maybe a low one, 0.3. And then I am going to, for every one of them, I'm going to randomly just to random. I'm gonna say random number 10 of them. So 10 of them, yes. No, this is not pop. 10 is not pop. 10 is actually the number of chromosome. Did I make a mistake earlier? Darts. This is population. This is not pop. This is actually 10 digits, not pop. Okay. So I randomly generate 10 numbers. I need to check whether there's one of it that is smaller than the crossover rate. So I will say cross random numbers smaller than include the equal 0.3. Then I will have a bunch of 0 and 1s. I have a bunch of 0 and 1s, and if I make a dot product, this is horizontal. So dot product, a vertical 1 to 10. Then I will have a bunch of numbers. Some of them are 0, and the smallest one, and sort them. After sorting, I shouldn't do dot product. Sorry, I should do dot multiply, no transpose. And then I need to sort. There will be a bunch of zeros. So the second one, let me call this temp. Second one will be the one that needs to be hold on, let me just make sure, stop right here and see what's going on. No, something was wrong. Rend. So after that, okay, 0, 0, 0, and 1. 1 basically is the first one. First one needs to do crossover. So we don't want to do crossover in the first one because it doesn't make sense. If we do the first digit crossover, that basically gives you the same results. So we should we should start from 2. We don't care about the first one. So we only randomly generate. No. From 1 is okay. But when we randomly generate, we should say 0, 2 to 10. No. Yes, the first one is always 0. We don't even need to do crossover for the first one. If that's the case, then we will never see 1. So in this case, we need to do crossover at the second digit. So I need to do unique. After sorting, after unique, I will second number will be. So if length of temp is larger than 2, then the second number is the digit for crossover. So the crossover ID would be the second. Let me show you. So I randomly generate a bunch of numbers. Some of them are lower than 0.3. I use this trick to get numbers 0, 5, 8, and 10. That means the 5th digit, 8th digit, and the 10th digit are smaller than 0.3. But I need to do crossover at the 5th one. I'll do it at the 5th one. So this gives me crossover ID, the 5th one. So what I'm planning to do is to if I do crossover, then 2 of them will change to new chromosomes. So later on, when I do selection, actually 1 chromosome could be crossover multiple times. Okay. So now you need to make a decision. Do you do that? Okay, let's do that. So whenever we exchange, just exchange. We forget about what's before. So when we do that, so the crossover rate shouldn't be too high, because you may be crossovering twice for each chromosome. So if we do that, that means the pth one and the qth chromosome are changing its body. So chrom selected the pth one, and starting from the crossover ID to the last becomes the body of the qth one. But if you do that, the tail of the pth one disappears. So we need to have a memory just called mem, equals to the tail of the P. So later on, the chromosome selected, the Qth one crossover ID to the end equals to the memory. Okay, so they change body. We can stop right here. Okay, the memory is, oh, sorry, there are 20 bits. That means this number needs to be 20. 20 bits, yes. Do it again. Okay, so memory is, okay, 17 numbers. That's the tail. And we can see, we can monitor this one. P and Q equals to what? P is the first, Q is the second. So chromosome crossover happened between first and second. So these two will change body. These two change body, and starting from, because there are 17 of them, so starting from the, I guess, the fourth one. So one step forward, you can tell the first one will have exactly same body as the second one, and do it again. The second tail becomes a different one. So the first and the second chromosome just change its body, exchange its body, its tail. Okay. Then this will be done repeatedly until a bunch of things happens. But I think the crossover rate is too high. Let me reduce to 10%. I don't want too many crossovers. So let's finish until here. So it will be a bunch of exchanges. You might be curious about what are the new points after this crossover. You can actually plot them out. We already figured out how to plot them out. You just need to convert them into values, right? So here you just can say selected, right? And I can call this selected, right? And what's my index? Index I. So I can use say for PQR, R equals to 1 to the size of X chromosome selected. I'm sorry, today I'm taking longer. If you cannot stay longer, you can go. I'm almost done. So this is converting back, then I need to do plotting here. Here just selected. But I'm changing a different color, use red. Okay? So let's see how those crossover, how does crossover change things? Mistake, typo. Selected. Oh, sorry, just this. Chromosome selected, yes. Okay. Chromosome selected. Why? IDR, not K, not not I. Run again. Okay. From here, you now see a bunch of new points. They came from the chromosome you had with those crossovers. But I'm not sure if we have. Let me change this to cross. I want to see if we still have some original chromosomes. Just do it again. Okay. So do we see cross on circle? No. That means we cross over too much. We basically cross out everything. None of the circles were kept. So based on the way I wrote, the crossover rate was too high. So let me reduce to maybe 5%, even lower. Do I still have? No, none of them still there. Maybe even lower, just 1%. Sorry. Okay, now you do see some. You see some chromosomes remains there. But this should be the best one. Actually, this one is also good. These two are good. So it's good that this one was kept. Okay? Because we know this is the best. Let's try again, see if the best point was kept. Over here you can see, okay, this one was kept. Which is good. And we do see some let me explain more on why we will have a point like this. This cross probably came from this point. What happened was maybe the x coordinate of this point, original point, has a slight change. Some of the bit changes from 0 to 1 or 1 to 0. So the position of the x coordinate changes a little bit. But sometimes it will have dramatic change because in the chromosome, each bit represents different portion of the value. If it's in the right hand side, it changes a little. But if it's at the left hand side, the very left digit, it changes a lot. So the randomness just change a little bit. Sometimes that change moves the coordinate a lot. Sometimes it doesn't move a lot. Okay, and also since we have a long chromosome, and sometimes we change bodies, right? If the changing digit locates in the x1 position, then the entire x2 exchange with the other chromosome. It's like you are sharing your x2 coordinate. You took the x2 coordinate of your neighboring chromosome entirely. So it's like maybe something good over there in other chromosome. You took it. And the reason why we want to do this fitness value selection is because we want to have as many good chromosomes as possible. Otherwise, those random process can easily dilute those good ones. That's why we need more of those. Okay? Just one last part, we need to do mutation. Mutation should be easier. For loop Q, let's use S. So for mutation, I need a mutation rate. Usually it's even lower. So here I need to generate this mutate, and then I need this exact same code. I need to find a digit for mutation. This time mutation, I didn't use it inside the code, sorry. I was using 0.3 all the time. That's why it was still high. So maybe it's too low. Let me try again. Just to check. It was 0.3, right? It was what number? It was 0.3, yes. I should here. I didn't use this setting that I used defined earlier. So if I use 0.1, I think it will be okay. Sorry, try again. Is it keeping the good ones? Yeah, I think those are good ones. Okay, that's fine. So here maybe 5% of mutation. If this happens, generate those. Now, okay. So I need this as well. If this happens, it's called a mutated ID equals to 2. We don't need to memorize. We just say the S1 and the mutation ID changes from 0 to 1. So what is 1 to 0, 0 to 1? Equals or? There's no equals or? Oh no, equals or. Okay, so needs equals or 1 and 0 goes to 1. No, not equals or. Or 1 and 0. No. How do I? Never mind. Let me write a stupid code. If let's use a switch. Switch this value, case 0, then change it to 1, and then case 1, change it to 0. I wanted to use one line, but I couldn't think of a good way. Okay, so that generates the mutation. So we can also try to plot out the result of the mutation. Exact same code here, but this time use a square and green color, maybe magenta. Okay, this should do it. Let's see. Okay, good. So how many mutation? 1, 2, 3, 4, and 5. 5 mutation. Many of them remain at the same position, at the crust. That means mutation didn't happen. I think it's good. I think this ratio is good. So once this is done, we need to make sure the results are feasible. So we need to do feasible check. Here, feasible check. And we need to check fitness value. Then we're done for the iteration. So the final check for new chromosomes. So over here I have all these chromosomes selected, the updated ones. Are they updated ones? Yes. So I'm going to plug in into the functions. At the end, I need to over here I already have this check, right? Yes. So I'm going to kill the bad ones. So I don't need the feasible flag anymore. I just need to directly kill the bad ones. So whenever invisible flag happens, I killed it. I killed it. So that particular one, basically the jth one, killed. I better not do it. I better use a flag as well. So whenever there's a flag, I need to memorize which one, like invisible memory. Invisible memory, okay? So if there's an invisible one, my invisible memory will remember which one is bad. So invisible memory and the J, the index, okay? So whenever there's a bad one, I keep a memory of it. So at the end, I could have, I need to say invisible memory equals to unique, because I may have duplicated ID in these ways of memory. Then those ID should be killed. So it's okay to kill them. Those ones kill. That kills them. Now we need to do feasible check. When I check, I only check the remaining ones. So I plug in those values. Then I will get all the I better clear Y because I used Y earlier. I clear them again, or I will just say, yeah, just clear Y. I don't want to. So at the end, those will be the fitness. Let's see what happened. Okay. Any bug? Okay. Invisible memory 2. Oh, something happened. Not the X chromosome. It should be the chromosome. X chromosome memory. So just X chromosome, not X. Chromosome selected. Chromosome, no X. Yep. Yes. Do again. So done. How many was killed? Invisible memory 2. But so many invisible. What happened? Should be more. Let me see why. Maybe we stop here. Sorry. The cross one, there should be a lot of J, yeah, index. So once we're up to here, I should have a bunch of only one. How come? Empty. Did I have typo? No. Let's do it again. Oh no, none of them. When I plug in the constraint, did I plug in the right one? Let me see. Ah, there's many of them. So I should say I only plug in one point. This is for plotting, so I should say R comma. Oh no, because this is like checking one at a time, So this should be inside this loop, inside the loop, checking one at a time. So it will be computed 10 times. So I can call this R. Sorry, this is taking a lot of time. Let me rename it CC. Or just add underline, just to avoid same coding as before. Once I have those, I can do this outside. That's fine. So I should be able to get a bunch of. Okay, what's wrong? Hmm? Cannot be empty. I think it computes this. Sorry. It's a vector. It's a vector. It's a vector. So like this. Sorry. Oh, there could be multiple of them. So I need to check one by one. Right. So they are vectors. So it's actually those are checking one by one. So I should use invisible flag. Actually, earlier it was right. Okay, only if all these satisfies, this particular one is invisible. So invisible memory will be outside of here, and then R. Yeah. So that will check. This goes to empty here. Okay, unique. Yep. You should do it. I have several of them. 10 of them, really? No. This goes to 0. Try again. How come they are all... sorry. If this equals to 1, then keep the keep it as the invisible memory. Yes. Okay, do it again. Almost done. So 6 of them invisible. 1, 2, 3, 4, 5, 6. Yes. Okay. I think the mutation rate probably too high, or crossover rate too high. It's okay. It's GA. GA does that. Okay, now we run until the end. I should have a bunch of Y's. They are no. I need to check. I didn't keep it as a variable, so I need to check fitness one at a time. So after so here I can, if I check feasible, I can also check the function value. When I kill the function value, fitness value as well. So when I kill the function value as well. Should be done. Okay, I should have a bunch of Y's. Mistake. Let's wait on. I shouldn't have clear Y here. I should clear Y outside. Clear Y. Yeah. That's why. No. The index is not. It should be R. Selected. Just plug in. Yep. Come on. Okay, finally. So I have 7 of them. Chromosome remains 7 of them. They are the remaining ones. So I only keep the best one. I need function value to be the smallest. So mean Y temp ID equals to mean Y. Okay, so this ID is the elite ID. That one will be kept, elite ID, and we will memorize the elite ones. So chromosome elite equals to the chromosome selected, and elite ID. That one was chosen. And just to show on the screen, we can We probably need to plug in again because we don't know what's the coordinate again. Just to show on the plot, we can convert it into x just to see what's the elite ID. What's the position of the elite point, and then we can say elite, and then we can plot it. Plotting is here. So for this special one, I'm gonna choose a circle, red, larger marker size, and then thicker lines. Okay. Should be the best one we found. Something was wrong. We don't need R. Okay, you're right. So just just elite. Should be. Oh, then this should be like this. Okay. Did we choose the best one? Yes. So originally we have circular ones. Those are the chromosomes. After crossover, we have the red cross. After mutation, we have the square. And then we have the best one among all these random search. So you can see this one actually was not from the original random search. Crossover and mutation somehow move our point to here by some randomness. Okay, actually this already, we know the solution probably is here. We are already really lucky. But how many times have we already plugged into the function? 35 times. So it's not cheap. It's not a cheap method. Okay, so it's already close to 1 p.m. Actually, for the next iteration, I need to come here and then do something to keep the best one and then continue to do it. I'm almost done. Let's do it. If you have to go. So in the next iteration, so I should say if exist variable. If this exists, that means we do have a elite. Then chromosome 1 equals to chromosome elite. Okay, and then chromosome. That's it. And then I equals to I plus 1. Right? Because I want to keep the elite one. And then for the rest, just randomly generate. And everything is the same. If everything is correct, it will does the magic. No. Life is not easy. Why? Maybe we need to clear some of the variables in the second run. Okay, let me check again. Because this is where I make a new chromosome. This is my chromosome index. I keep the first one. This is where I clear the chromosome. And then I keep the first one. So starting from the second row, repeat until the size of chromosome. Yeah, sure. Let's check here. In the first time, chromosome is empty. Second time I should have some chromosome. Actually, I should check. I should stop here. Okay, this is already second iteration. Chromosome is empty. Step forward, so I have one good chromosome. This generate second chromosome. Yes. So nothing was wrong here until maybe we stop here and see. Okay, so I have finished. Okay, here is wrong. I should not have this many. The keep. Okay, second time. Why is that? K and 1. Oh, so when I think later on I use Y. I shouldn't use Y again. I should use a different name. Let's use capital Y. They screw up what I had earlier. Okay. Okay. First time, good. Second time, I should have 10 values. Okay, good. So should be okay. No, still some problem. So let's first time again. First time. Okay. Throw away these. Why? Why? Yes, I now have this Y. Okay. Step. So these are the capital Y's. EDID. Step forward. EDID. Best one is the third one. Chromosome select. So I do have a chromosome best one. Yes. So nothing wrong with these, I guess. Chromosome elite. Yes. So go. So I can plot. Okay. This is first iteration already. Oh. Okay. Second iteration. Now we have we want to delete this many. Go. Okay, Y. Okay. Keep going. Okay, so this was the first iteration, this is second. So I guess earlier, let's see. First iteration. Okay, maybe when this is happening, all the yeah, everything was invisible. That's why. That's why everything was killed. So here, if we kill everything, then so if invisible memory, if okay, that means so if in the length of invisible memory equals to the number of chromosomes, so pop, then we don't need to do anything. We do nothing. Otherwise, we will do this. So that means if everything becomes invisible, we still keep the elite. We jump to the next iteration. Because that means in this generation, everything was killed. Now we can turn off some of the display, because we don't need to see so many. We just want to monitor the elite ones. Okay, this will only show the elite ones. Oop. Okay, so sometimes the dart. What's the dart? Dart. When was the ID here? How come it's not valid? Oh, by the way, if we have the elite ones, we shouldn't mute. We should keep the elite ones. Otherwise, we are going backward. If we didn't find a better one, so when we mutate and cross over, we should keep the elite one. Okay, so after everything, I want to keep the elite one. So before feasible check, this is final check of the final chromosomes. So I want to add one more because we already did some crossover mutation, something crazy. So let's just put the elite one back. Chromosome selected equals to chromosome selected and chromosome elite. That will make our elite one back. So did we use population here? No, so we will have 11 of them. So then here is plus 1. I will have 11 of them. So supposedly I won't be going backward. I will be always improving. Typo. Chrom elite Because in the first iteration, there's never a Chrome Elite. So if exist Chrome Elite, then you keep it. Otherwise, you don't need to add one. Now you can see it's always improving, but something was wrong. I don't know. Sometimes when I compute index, something was wrong. When I throw the darts, maybe I do it again. That line gives me mistake. But you do see it's improving. It moves closer to the optimal. However, this line has some mistake. I need to go back and check. I don't know where. But it's there. You just need to debug here and there. When I do programming, there are some careless programming. Like I use the same variable names, and then here and there are some issues. But you get the idea. You can tell how randomness helps GA to find the solution. Even for here, I think we're up to the 4th iteration, and the function call is already 300. That's a lot. The maximum trial of all combination is 2 to the power of 20. Right? All possible combination. So it's actually 1 million possible combination for the entire chromosome set. If you can find a solution within 1 million times, you are doing better than exhaust search. Okay, so over here we try. I think we stopped at 4th iteration. I think because I see 4 points. So it's like every iteration it averagely needs 100 function calls. So if I try 100 iterations, then in total I need 10,000 function calls. But the possible combination is 1 million. You are doing way better than exhaust search. That's the power of GA. But as you can see, compared to all other gradient approaches, this is much more expensive. When do you use it? Like I said, for discrete problems, for problems you just cannot take derivative, cannot do gradient, maybe randomness based methods probably will be the only way. Okay, that's it for today. I'm sorry I used 1 more hour. Thank you guys for coming here. I will upload. I probably will fix the code and then upload, so you can check later on.